Hello, thank you for joining. As part of the ERA and PigRegen funded project, we have prepared short presentations with the main outputs from experimental trials. Most of the research was done by the monogastric research group, and most of it was conducted at Acknowledgments Pig Farm in Hillsborough. For the current presentation, I'll be presenting it myself. My name is Dr. Ramon Muns from the monogastric research group, and I'm going to be talking about the evaluation of different foster strategies to deal with large litters. So as we know, genetic selection during the last decades have been pushing towards sows producing large litters. So that this is resulting in that the number of piglets born per sow it outnumbers the number of functional teats of the sow. So therefore, that requires some extra management to deal with large litters. So we are talking of 14 and above live-born piglets per sow. Cross fostering within the first 24 hours of birth has been a useful tool, a useful management so far. So, for example, litter equalization, or as we call it, fixed litters by number, in which we would just transfer piglets or foster piglets until we get the desired number or litter size, or litter standardization, which are also called fixed litters by size, in which we would cross-foster piglets until we get a litter of small piglets, a litter of big piglets, etc. So these practices are the ones that we have been using for years. But now, with high prolific sows, these might not be enough. One strategy that has been gaining popularity is the use of nurse sows. In which we can have two main systems, the one-step nurse sow system and the two steps. The one-step consists in maintain a sow that is being weaned with 21 or 28 days of lactation. So this sow is retained in the farrowing house, and when the new batch comes in and it farrows, this sow is given the surplus piglets with 1 day of age. The two steps system, we use an intermediate sow. So we have the 21 or 28 day lactation sow that we keep in the farrowing room. Then this sow is given 7-day-old piglets. And then the sow, 7 days into lactation, that is empty as a result of this movement. Then this sow is given piglets of 1 day of age. So we'll try to see it in a more graphic way because it's complicated to explain. So here in this figure, we have on the left we can see the one-step nurse sow system. So the black sow would be the sow just weaned, and then it receives the surplus piglets from the newly farrowed sows. On the right, we have the two steps in which we have this intermediate sow in between. And in these cases, usually is the sow that is moved into a new batch. So we have an older sow or a sow that is already 21 or 7 days into lactation, which is moved into a batch that is just being farrowed or 7 days into lactation. Another system that is gaining popularity to deal with these large litters or these surplus piglets is the use of artificial rearing, or as a commercial name, rescue deck, which consists on removing piglets from a sow at 14 days of age, and they are transferred in this specialized enclosure or so-called rescue deck. And this rescue deck can be located either in a separate room or above the sow's farrowing crate. And then piglets are fed artificial milk and crib feed. So the objective of the present trial was to study the effect of different foster strategies on the performance of piglets during lactation and post-weaning. These foster strategies are a combination of nurse sow and rescued egg. So this trial was conducted in a commercial farm here in Northern Ireland, a farm with 1,500 sow herd. For the trial, we used 63 sows and approximately 790 piglets. And this farm worked in a one week batch system, weaning at 28 days of lactation. So we identified 4 treatments. The control treatment, in which piglets were left with the biological sow for the whole lactation, so no movements here. Day 1 treatment, in which a whole litter was fostered at 24 hours into a 7-day lactating sow. So a 24 hours old litter was moved to the previous batch, in an empty sow that was 7 days into lactation. The day 7 treatment, in which a whole litter with 7 days of age was fostered into a sow of 40 days into lactation. So the whole litter was moved into a previous batch and into an empty sow that was 14 days into lactation. And then the last treatment, the rescue egg, which was a whole litter was transferred at 40 days of age to this artificial rearing facility. To try to understand better the treatments, we'll see this graphic or these pictures. So here you can see pictures of the rescue egg to get an idea of how they look like. So from a sow 14 days post-farrowing, we moved the whole litter into a rescue egg. Then this empty sow is filled with a whole litter of 7 days of age, and then we have this sow 7 days into lactation that is empty, and this sow will receive the surplus piglets, a whole litter of 24 hours of age. And then in this empty sow of 24 hours post-farrowing, this sow will receive the surplus piglets of that batch. So that resulted in the treatments day 1, day 7, and rescue egg, which, since the litter was moved forward, so was moved to a previous batch, these piglets were weaned at 21 days of age. And then the control treatment, which, since they weren't moved at all, they were weaned at 28 days of age. It's important to note that all the movements, all the litters that were moved, they were always selected big size litters, so litters in which the piglets were big or heavy. Then we measured individual body weight, so we have the piglets individually identified with ear tags, and we measure weight, birth weight, weaning, either 21 or 28 days of age based on the treatment, and then body weight at day 40 of age. We monitored all these piglets and litters transfers to nurse sows and rescued eggs across 4 batches, so allowing us to obtain enough litters or piglets per treatment, and all the data was analyzed statistically. The results here in this graph, we can see that when we compare the birth weight of the piglets, because the way we proceed is in 4 consecutive batches, we weigh as much piglets as we could or litters as we could at birth, and then we waited until the treatments were created by the farm staff. Due to there was initial bias, and we can see that among treatments there was a slight difference on birth weight. Nonetheless, statistically, we could introduce these initial variants, so is accounted for in the subsequent analysis. Therefore, it's not influencing the results that we will be seeing. So at weaning weight, as expected, the control piglets were heavier because they had 28 days of age, whereas the day 1, day 7, and rescued egg, they had 21 days of age, so therefore they were lighter. But when we compare these 3 treatments, the red bar is day 1, green bar is day 7, and the purple is rescued egg. When we compare these 3 treatments, we can see that day 1 and day 7, they had similar body weight at weaning, whereas rescued egg were lighter. So moving the piglets into a rescued egg, it seemed to reduce its growth and its weaning weight. However, interestingly, when we weigh the pigs again with 40 days of age for all treatments, we see that there are no differences among treatments. So they all had similar body weight. And it's very interesting that the rescued egg catch up on growth with the other treatments. And even numerically, it's even higher, even though it's not statistically different. So that makes sense because piglets move into a rescued egg. They go to a partial weaning, so they are separated from the sow and fed artificial milk and crib feed. So that creates a stress, a partial weaning, that could explain this impairment of growth during the next 2 weeks. Whereas when we do the full weaning process, rescued egg piglets cope better because they have already been separated from the mother. So for them, it's just a movement of facility and a removal of the artificial milk. So they cope better with this change, while all the other piglets, they go through all the stressors at once: removal from the mother, change of diet, change of environment. So that's why we think we observe these changes in body weight. So our results, even though our trial it's not comparable to other published studies, and there aren't many published studies on the use of nurse sows, but our results somehow agree with what it's published. So what we can see in studies published is that the growth and weight of piglets at weaning did not differ among pigs left with a biological sow or pigs transferred to a nurse sow. And in the literature, we also see that the 2-step nurse system is preferred or recommended compared to 1-step, which in our case, it would be more similar to a 2-step than to a 1-step system, the treatments we compare in our trial. And as a concluding remarks, so move litters forward in a 1-week bite system, either at 1 or 7 days of age, does not seem to impair pre or post weaning growth. And the use of artificial rearing or rescued eggs reduces pre-weaning growth, but those animals are able to present compensatory growth after weaning. And it's very important that to always move the big pigs, the heavy litters. I wouldn't recommend to move litters or piglets that are light or small. And then after this trial, we had some questions or aspects that we couldn't study in our data. Like, for example, we don't have information of mortality because we couldn't record it properly in a commercial setting. It would be also very interesting to follow these animals to slaughter, which we couldn't do. And also, since we're moving animals from a biological sow to a foster sow or to a rescued egg, changing the type of milk that the animals receive for their age, it would be interesting to see if there are any impacts on immunity, health, or also in behavioral aspects of the animal. So all these are topics that I hope in the future we'll be able to study. Finally, just the acknowledgments to the Data and Picrogen for funding and co-founding this project and this work. A big thank you to Trevor and his team that made possible, allowed us to conduct this trial on the farm. And also a thank you to Chagas, because Chagas participated in this trial. So they were involved and they collaborated to make it possible. Thank you very much.